This experiment is focused on concepts of precision and accuracy, as well as introducing the idea of precipitation reactions and average heating rates. And really, it's meant to be an introduction to making measurements in the lab and how to think about how accurate your data is and how precise your data is. So, let's begin by a discussion on precision and accuracy. These are both qualities of data that are important to discuss when thinking about how good the results that you obtain in the lab are. And so let's imagine we are trying to measure a sample that we know its true value is a 1.7500 gram mass. And we have a series of different balances, and all of these balances can go to 4 decimal places in terms of the measurements they're able to make. So we place the sample on these 4 different balances, and we get the data shown in this orange box. As you can see, these values are pretty spread out and do not seem to be very close to the actual true value that we are attempting to measure. But one way that we could kind of consider this data is to take a mean value of all of our different measurements and see if it's close to the true value. So I took the mean of this data and found a value of 1.5519 grams. So the question is this value that we obtained as a mean of all of our different sample measurements accurate? And another question is the data that we obtained precise? So these are 2 different questions. They often get confused, but hopefully this discussion will help to clarify what we mean when we say that. So let's imagine we have measurements that are spread out in a pattern that look like, here I'm going to use pink for this, that look like this. All of these data points are not very close together, and that would represent a low precision sample data collection. And so really precision is looking at how far apart all of your data points are. On the other hand, we could have a sample that the data points we measure are close together like this, and that would represent a much higher precision. So your data points themselves are close together. All right, so that's the idea of precision, whereas with accuracy, it has to do with how close your measured values are to the actual true value. And so the X here is representing the true value that we are trying to measure. Maybe we already know what it is from the literature or so on. But if a sample is not very accurate, the sample measurement data points could be way off from the actual target value. Whereas if there is high accuracy, the data points could be very close to the true value. So that is sort of a conceptual difference between precision and accuracy. Now, if we want to actually put some sort of number to represent how precise and how accurate data is, we can calculate the standard deviation to show essentially the spread of the data points. And that is calculated using a formula, and Excel can do that for you. Also, if we want to look at accuracy in terms of a number, we can calculate the percent error in the measurements, or the percent error in the average of the measurements, which is probably more likely. So this tells distance of mean from true value. All right. So hopefully those kind of calculations make sense with the conceptual explanation. But percent error, there is also a formula for that, measured minus true over the true value times 100 should give you the percent error. All right, so another aspect of this experiment, well, actually, I'd say before we move on to that, we could return to the question of, was our value that we obtained for the mean accurate? And by looking at the distance between these two values, it seems like it has a fairly large percent error, meaning that it's fairly off from the true value, but we could actually calculate that to determine how close the two are. Another question we asked was, how is this data precise? And I would think that if you were to measure this sample on 4 different scales and get this range of values, you'd probably think they're not close together at all, and you'd think you're doing something wrong. So most likely this is not precise at all, but we could calculate the standard deviation to determine that relative precision. All right. So the another part of the experiment that you'll be conducting is you'll be heating water sample using a temperature probe and measuring temperature over 10 minutes at 5-second intervals so that you can see how quickly the water is heating on your hot plate. And most likely you'll observe the temperature increasing up to a point where it begins to level off, and that is the point where the hot plate is not really heating any longer. It's maintaining the same temperature. And you'll have a whole bunch of data points that you've collected across time, and you can kind of plot them in Excel. And it's just important to remember that your axes should have units. So if you're measuring in Celsius, you should definitely indicate on your graphs what the units of your measurements are. So temperature in Celsius, time in seconds. Now, one thing we could consider in our measurement of heating is what is the average heating rate. And there are different types of lines that you could fit to your data depending on what type of analysis you're doing. Later on, when you get to the second chemistry course or in 1310 later in the semester, you will look at kinetics, and there will be different types of functions that we would fit to our data points. But most often, we will fit a linear function to our data points. And so I'll draw that here. Essentially, when it's fitting a linear model, it is trying to minimize the distance between the points to the line and still maintain a straight line. And so the slope of that line is often important for whatever analysis we're doing, but we essentially get out of this a function of y equals mx plus b, and that's generic, but whatever is our y-axis would be the variable that is plugged in for y here. So temperature is our y-axis. Whatever is our x-axis is plugged in as the variable for x. So we have time, and then the slope is going to be y over x, which is temperature over time. And then, of course, we have b, our y-intercept. So b basically represents when we're at time 0, what is the starting temperature in this example. But the line that we fit here, the straight line, is essentially going to represent the average rate of heating of our sample. So we could consider that slope as the rate of heating. Okay, so that is part of the experiment. You'll be collecting this data digitally and able to plot it and your plot will appear in your post-lab assignment. But there's one final portion of this experiment that we'll be looking at, and specifically this is solubility and how that relates to whether or not a sample will precipitate. And so we'll be combining a wide range of different samples together, and in this experiment you'll be looking at whether or not a precipitate forms. And so a precipitate could look like your solution has become cloudy. It could look like you actually see solid particles, but both are valid observations that a precipitate has formed. If it remains clear, you would say that there's no precipitate, and therefore whatever was formed must be soluble in water. So let's think about this concept of solubility and precipitation reactions in terms of what's known as a double replacement reaction. In this type of reaction, you take two salt compounds that are composed of a positive and a negative ion. So in this reaction, A would be our positive ion, B would be our negative ion, C would be another positive ion, and D would be another negative ion. And when they're written together like this, they are salts. And these are just very simplified. A lot of times you'll see subscripts to represent the numbers of ions that are paired together to give you an overall neutral charge in that salt. But let's just assume here everything is a +1 or a -1 charge, and that's why we have no subscripts written. When you mix A, B, and CD together, they will essentially swap partners so that the positive and negative opposite components are able to combine. So A will form a compound with D, and B will form a compound with C. And actually I wrote this in the wrong order, so let me erase this. It should be BC, if we're going to be proper about what order you write things in. All right, so this is a double replacement reaction, and this is essentially what's happening whenever we mix together two salt solutions. They're dissolved in water, and if they're dissolved in water, that means they're inherently soluble. But when we mix them together, we may or may not get a soluble mixture. So when we want to make predictions about that, or even understand what might be happening, a useful tool are solubility rules. And so I made up a couple of generic solubility rules to kind of help us think about this generic reaction. So often you'll see rules that say that something is always soluble except in certain circumstances, or you might see that other types of ions are always insoluble except in certain circumstances. So let's say A+ ions are soluble pretty much always, except when they're combined with D- ions. All right, so that's one rule. And let's say another rule is B- ions are always insoluble, except when they're combined with A+. So if we were to follow these two rules, we could think about the compounds in our overall reaction to try to decide which ones might be soluble. So A+ ions are soluble. So the two compounds that have A+ in them are AB and are AD. But the rule says this is not true when it's combined with D-. So it's not soluble when it's combined with D. So that means that AD will form a precipitate because it is insoluble. So we will see solid appear. That may be cloudy, that may be physical particles falling to the bottom of the test tube, but that solubility rule leads us to that conclusion. All right, the second rule we're given says that B ions are insoluble, so that means that they will not dissolve usually, except when they are combined with A+. So that means that, again, this AB molecule or AB salt is going to be soluble because A is soluble and B, when combined with A, is also soluble. So that's fine and dandy, but what we really want to know about is this BC molecule. Oh, I was actually right before. I don't know why I changed this. This should be CB. All right, so here we go. CB. So B is usually insoluble, and the only exception it gives us is with A. So the conclusion we would get to is that this would also form a precipitate. So that means that we would see a lot of solid forming when we mix these two AB and CD solutions. And in this experiment, you'll be using an unknown solution and trying to draw conclusions about what is actually present in it. So you'll want to try to figure out what is the cation and what is the anion, and you have a limited set of possibilities, which are also given solubility rules. And you're given a set of known solutions to react them with. So your task will be to systematically react your unknown with the known solutions and identify patterns. So you'll have to carefully track what results you're observing. Are you seeing a precipitate? Not seeing a precipitate. And you'll have to draw some conclusions about it. Is that precipitate caused because of the anion in the known solution or the cation? And multiple different tests can allow you to draw those conclusions. But it's kind of a holistic analysis of your set of data with the information you're provided.